Good morning, good afternoon, and a good evening to everyone present here today. I would like to welcome you all to the first talk of our Life Sciences webinar series this year. My name is Sahil Gulati. I'm the Applications Manager of Life Sciences at Gatan. Now, before I introduce our speaker for today, I would briefly go through the question submission process. You can type in your questions in the question panel of the GoToWebinar cast window, shown on the right-hand side of this slide. We will try to answer all your questions immediately after the talk, depending on the time remaining. If we do run out of time, then your questions will be taken offline, where the answers to your questions will be sent by emails. With that, I would like to introduce our speaker. I welcome Anthony Schuller, who is a Helen A. Whitney Postdoctoral Fellow in Nogales' group at University of California, Berkeley. A little bit about Anthony. Anthony first became interested in structural biology as an undergraduate student at the University of Pennsylvania, where he started working and was a part of a team that solved the first high-resolution structure of the telomerase reverse transcriptase enzyme using X-ray crystallography. He pursued his Ph.D. in Rachel Green's lab at the Johns Hopkins University, where he studied the fundamental mechanisms of eukaryotic protein translation and mRNA quality control. At the end of his doctoral work, he became interested in cryo-electron microscopy technique, particularly in how cryo-electron microscopy could be applied to complex samples, such as an entire cell using cryo-electron tomography. To pursue this work, he moved to MIT and started working with Thomas Schwartz to use a combination of cryo-FIB and cryo-electron tomography to study human nuclear pore complexes inside the cell. Before moving to Ava's lab at UC Berkeley, Anthony was with Thomas Schwartz for 3 years, and the work he conducted in those 3 years was recently published in Nature Journal. And this is the work he'll be sharing with us today. The title for today's talk is Context Matters. Cryo-electron tomography reveals the impact of the cellular environment on nuclear pore complex architecture. And with this, I will leave the podium to Anthony.
Thank you so much, Sahil. Thank you for the invitation. I'm excited to share a bit of our work today with everybody on the call. So thank you for the introduction. I'm going to give just a short additional introduction because I like to show sort of the scale at which I've approached structural biochemistry in my career. As you mentioned, I started with crystallography at UPenn in my undergrad studying telomerase, and moved to Johns Hopkins studying a larger macromolecular machine, the ADS ribosome in yeast in Rachel Green's lab, and started to get interested in cryo-electron microscopy and cryo-electron tomography, and moving up in scales to even bigger and bigger things. So if we make this appropriate to the scale of these molecules inside the cell, I just like to show how big the nuclear pore complex that I'll be talking about today is in comparison to these other macromolecules that I've worked on in my scientific time. So today, a brief outline for what we're going to discuss. I'm going to start with a short introduction to cryo-electron tomography and what cryo-electron tomography has done for accelerating EM sciences in complex samples. And then I'm going to tell you about some biology that I was interested in, am interested in, is how the nuclear pore complexes mediate nuclear and cytoplasmic transport. I'm going to bounce back to technology and tell you about our use of cryo-focused ion beam milling and how that sort of opened a new avenue for biological exploration, and sort of end on combining the two, the biology and the technology, to show how we sort of unveiled how the nuclear pore is impacted by the cellular environment. So this audience here probably knows very well that we had a Nobel Prize in Chemistry in 2017 in the field of cryo-electron microscopy. I got into cryo-EM shortly before that right here during the resolution revolution at sort of the biggest maybe inflection point when resolution started to get better from all of the various equipment that was coming out, direct electron detectors primarily. And this is the point where I was in my Ph.D. and looking into a postdoc and trying to figure out what I wanted to do. So this is what really inspired me to get into cryo-EM. And you know, in 2015, here we were at the average resolution, or the highest resolution was somewhere between 2 and 4 angstrom, and now cryo-EM is rapidly approaching you know, near atomic resolution with these 2 papers that came out last year from the Stark and Schuller groups with some fancy microscopes that have a bunch of extra correctors and some fancy new detectors. But you can see that they're really getting to as close to atomic resolution as cryo-EM has gotten. So this is really, really exciting, and I'm really excited to be a part of this growing and expanding field that's pushing the limits of structural biology. So as a means of introducing cryo-ET, we obviously have to start with single particle cryo-EM. This is what sort of is the gold standard for solving protein structures by cryo-EM right now, is we take images or micrographs that contain tens to hundreds to thousands of particles in varying orientations, and we classify those orientations and reconstruct a 3-dimensional structure from those images. Now, the primary difference between single particle EM and cryo-ET is that cryo-ET, or cryo-electron tomography, involves tilting the sample to collect a series of micrographs and then reconstructing from back projections of the tilts. So you have an inherently thicker sample that in the electron beam gets tilted at various angles, and then the images collected at those tilts are back projected to create a 3-dimensional volume of the original sample. So what this looks like in video form, I got this from a friend at Thermo Fisher. Inside the TEM, you insert your sample on this grid here. That green beam is the electron beam, taking single micrographs through various tilts of the sample. And at the end, you have a series of tilted micrographs that you can combine using computational resources to make a 3-dimensional volume for studying larger samples. In this case, this is a whole cell here. So the benefits to cryo-ET, primarily that you can visualize thicker samples, and that allows you to identify proteins inside of a more native environment. We're not purifying them biochemically and looking at a grid that is you know, nearly homogeneous for a single protein, but instead purifying an organelle or not purifying anything at all and looking at proteins in their native environment. And then to achieve higher resolution with cryo-electron tomography, we employ a technique called subtomogram averaging, which was nicely illustrated by John Briggs in this schematic. So you have a 3-dimensional tomogram, and you can essentially identify your particles of interest that are aligned more or less randomly inside that tomogram, and pull them out into boxes, or cubes, more or less. And then you can align these to a reference, and you can see the particles are now all aligned in the same orientation. And this averaging computationally creates a new reference. You can iteratively perform this procedure, and at the very end, you come up with a more high resolution structure for your protein molecule of interest. And this has been employed for years. This is one of my favorite recent examples from John Briggs' group, studying something that's very important to our life currently, the SARS-CoV-2 virus that causes COVID-19. So they use some subtomogram averaging to solve structures of the various states of the spike protein. And this way, cryo-ET can afford you high resolution structure. Oops, I'm sorry. So in this way, cryo-ET can provide you high-resolution structure, but it can also provide you context in that you can see the various forms of the spike protein on a single viral particle. So from combining the subtomograms, you can get a high-resolution look at the spike protein, and then by mapping that back onto the three-dimensional volumes, you can understand the context of where different forms of the spike protein are on the viral particle. And it's this combination of structure and context that I think is the most important and most interesting thing that we can get out of electron tomography. So going back to our outline today, I'm going to move next into telling you more about some biology, telling you about the nuclear pore. So in eukaryotic cells, the nuclear pore complex is this massive proteinaceous macromolecular assembly that mediates nuclear and cytoplasmic transport. So anything in the nucleus that needs to get out that is larger than approximately 30 to 40 kilodaltons needs to be actively transported out of the NPC into the cytoplasm, and anything that is in the cytoplasm that needs to come into the nucleus needs to go through the NPC as well. So this massive structure is composed of many, many, many proteins, more than 30 different nucleoporin proteins. And comparing yeast and metazoa, you can see that even across species, while the major assembly is conserved, there are additional proteins that can be found in higher order organisms, such as humans. But for the sake of today, we're not going to focus on all of the specific nucleoporin proteins. What I'm going to do is more or less break down the NPC into three primary structures: the cytoplasmic ring on the cytoplasmic side, which contains 16 copies of this assembly called the Y complex because of its distinctive Y shape. And the cytoplasmic region also contains these set of nucleoporins. In the nucleoplasmic ring, you also have 16 copies of this structurally important Y complex and some other nucleoporins that are specific to the nuclear side. And then sandwiched in the middle, we have the NIC96 or NUP93 complex, which forms the primary central channel for the nuclear pore. And inside that central channel are the FG-rich nucleoporins that actually perform the transport properties. I wanted to just give this right now as a general overview, and we'll come back to this this three-ring structure later. So cryo-electron tomography has been used a lot in the nuclear pore field, even back to 2004 in the Baumeister and Medaglia groups, where Martin Beck was the first to employ cryo-ET to solve a nuclear pore structure. And this is in dictyostelium nuclei. You can see in these micrographs, they're imaging just the very edges of the dicty nuclei, and you can see with these arrows are pointing out the side views of nuclear pore complexes. And using subtomogram averaging, they were able to identify that the NPC has a three-ring architecture. They could see the cytoplasmic ring on the cytoplasmic surface of these nuclei. They observed a nuclear ring with this nuclear basket-like structure at the nucleoplasmic side, and then the inner ring architecture was also readily visible from these subtomograms, even in the early days pre-direct electron detectors, so back in 2004. And now, to show sort of a more recent, a higher resolution NPC structure from subtomogram averaging, Ohad Medaglia's group also employed this with Xenopus nuclear envelopes. And from these samples, you can see all of these arrows show the various NPCs in the nuclei. They can get approximately 20 angstrom structure, showing the details of each of the three rings in much greater detail than before. So this is really exciting from the perspective of what some subtomogram averaging can do for all sorts of samples, and especially the nuclear pore, which is what I was interested in. So, the motivation for my project in Thomas's lab was really to study the human nuclear pore complex in situ. And I've already given away the tagline here using Cryo-FIB. I probably should have deleted that before. But previous high-resolution structures all came from semi-purified samples, like I just showed you with the Xenopus nuclear envelope. And what I mean by semi-purified is cells were biochemically lysed, and the nuclei were disrupted because the nucleus is too thick and in many cases too dense with DNA to be visualized directly for cryo-electron tomography. So the cells were essentially broken open, the nuclei were broken open, and then nuclear envelopes were laid down on those grids for imaging. But my curiosity was really like, what do the nuclear pore complexes look inside cells that haven't been disrupted? Could these techniques of lysing and dissolving the chromatin inside the nucleus be impacting our understanding of nuclear pore structure? So, I mentioned the benefits of cryo-electron tomography. It can be used to visualize thicker samples, such as whole organelles or even small cells, and that leads to identification of proteins inside their environment. But there are also a lot of challenges with cryo-electron tomography. So samples can be hard to vitrify depending on how thick they are, and we have a solution for this in some cases. You can use high-pressure freezing to freeze large multicellular organisms or organoids. Samples that are thick can also be really noisy in the TEM, and one potential solution for that has been the Gatan imaging filters. We use the bioquantum imaging filter in our study here. And then, the third challenge that I already gave away the punchline to, was that samples can just physically be too thick to see anything. And one of my favorite depictions of this comes from a paper that Rebecca Thompson put out. And it just gives you sort of an idea of what TEM images or micrographs look like with varying scales. So proteins down in the bottom right here, you can see the individual protein molecules really well. Viruses you can still see quite well. Liposomes, even some of these microsomes you can see, but you can tell with a bacterial cell, you can really only see the details of the periphery on this bacterial cell, and the eukaryotic cell is just total blackness. So this is clearly too thick to visualize anything inside the cell. Maybe the periphery of the cell could be visualized, but anything in this major 90% of the cell here can't be seen because it's too thick. So the solution for this that had sort of been presented in the field is to use cryo-focused ion beam technology to create thinner samples for TEM and cryo-ET imaging. So while I was at MIT, we actually were blessed with two of these technologies. We had both a Zeiss instrument and a Thermo Fisher instrument that were capable of doing cryo-FIB and SEM imaging. And each one had some benefits. The Zeiss instrument was really good at the detectors for SEM imaging were really good, so that you could sort of see the samples you're making as you're making them, and Zeiss has sort of infinite control, so you could be very much in total control of your sample making. The Thermo Fisher approach had a colder stage, which was really nice for the lamella, and I would say the software was also and start making lamella right away. But it was really great to sort of interface with both of these technologies at MIT to make samples for this work. So how does this work? So essentially in these instruments you have an electron beam coming down from the top, and you have this ion beam coming down from a certain angle at the side there. And by using the two in the electron beam, the SEM imaging, you can see what you're seeing. And the FIB beam, the ion beam, is used essentially to ablate cellular material and create these windows into the cell. And so more or less, you draw these patterns onto the cell, and you tell the instrument where to shoot the ions. And it does as you tell it to do. This is very oversimplified, but I like it no less. and you can see you create these small, thin sections of a native cell that has been frozen, and then these sections can then be imaged in the TEM and tilted for cryo-electron tomography. So in my hands, what this looks like is we have grids, TEM grids that are coated with, these are DLD1 cells, human DLD1 cells. The grids are not treated in any way. They're just made hydrophilic and put into tissue culture dishes. Cells are laid on top of them, and you can see the cells adhere all over the place on the grid. And what we do is we identify cells that are in the middle of these square mesh, because then that allows us to tilt the resulting sample to high tilts for electron tomography. We zoom in on the cells, and in the case of this cell line and being interested in nuclear pore biology, we could actually see the nucleus directly in the FIB beam. Hopefully you can appreciate that these cells look a bit like a fried egg, and that darker region in the center is actually the nucleus of the cell. So we would just hone in on that region and then create these thin slices. For example, this one on the left here, which is obviously my best looking lamella that I ever made. But we create these thin samples. They're somewhere between 150 and 200 nanometers thick. And then we would image these in the TEM instrument. So if you image this sample directly without tilting it, this is the resulting micrograph. And you can start to see in this micrograph that we have biology here as we would have anticipated. So this stretch of membrane running through from north to south is the nuclear envelope. You can start to see mitochondria out at the top right. And there are some other features. I didn't pick the best one here and gave you even an ice particle that's on top of this lamella. But I'm going to walk you through the steps of reconstructing this tomogram so that you can maybe appreciate why reconstructing tomograms is important in the first place. So this is the same image I just showed you. This is a single micrograph on the left here. That's perfectly parallel to the electron beam. So in collecting this data, we perform a tilt series, and on the right is the raw tilt series from this exact same sample. So it's a little bit jumpy, and that's just because the collection software tries to track where it's located in XY positions, but it's not perfect. It's pretty good, but it's not perfect. So we take this raw tilt series and we align it computationally. And you can see now that things are pretty stable, and in some of the views there's a little bit cut off here and there. And that's just because everything now is better aligned in XYZ through the tilts. And then the next step is taking this aligned tilt series and reconstructing the three-dimensional tomogram. So now the video on the right is going to play a little bit slower, but you have to imagine a three-dimensional volume that we're moving through in Z slowly. And hopefully you can see here in the nuclear envelope section, these regions of fuzzy gray density that appear, those are nuclear pore complexes. On the right, you can see that mitochondria appears and disappears. Hopefully you can appreciate in the cytoplasm as well, there are all these black polka dots everywhere. Those are eukaryotic ribosomes. There are microtubule filaments running through here. It's just incredible how much you can see in these tomograms when you reconstruct this three-dimensional volume. So in this slide here, I'm comparing on the left what you get from a single micrograph with these thicker samples, and on the right what you're getting from the reconstructed tomogram. And I'm hoping that this really shows you that cryo-ET provides the cellular context that you don't get with single tilt cryo-EM. And in these tomograms, we can then do fun things like, in this case, I started to manually segment this tomogram just to sort of show the different molecules that we can identify. So these orange segments are microtubule filaments. This pink stuff here is endoplasmic reticulum because there are ribosomes bound to it, and there are other interesting features. Looking into the nucleus is a really fun thing to do if you have lots of time on your hands. It's really hard to identify things in the nucleus with all of the DNA there, but you can really start to pick out nucleosomes, and in some of our thinnest lamella, start to see connectivity between the nucleosomes, which I think is a really exciting avenue for future biology and future electron tomography. So in the data set that I collected for this work, I spent, and I included these figures. People ask me quite often, but this was early days of Cryo-FIB. So I spent 46 days doing Cryo-FIB, which gave me about 160 lamella, which means I averaged 4 good lamella a day. I was making many more than 4 per day, but some of them would be, too thick to image, or they wouldn't give me nice tilt series. And so that's this average of 4 good means I collected a bunch of tomograms from 4 lamella that were made each day. And so I spent 26 days with the Cryos collecting over 130 tomograms. We used the Thermo Fisher Tomo5 software to collect these tomograms in batch, which was pretty good at the time. And I understand that they've made some improvements to Tomo5 for batch collection, which I think are really exciting, and I would really like to try those out. And so for identifying the wild type NPC structure, we used 194 nuclear pore complexes collected from 54 tomograms. And then these were the statistics for the dose and the tilt steps that were collected for the tomograms on the Titan Cryos G3I with the Gatan K3 and BioQuantum GIF with a 20 EV split. And some of much of the raw data was so nice that we could even make out the 3-ring architecture of the NPC. So this is a binned micrograph, and you can hopefully see on your screens the cytoplasmic ring indicated here with this arrow, the nucleoplasmic ring here, and the inner ring. So this is one single NPC in the cell with a binned micrograph, and we're able to make out the features of this macromolecular assembly. I think this is really, really, really awesome to move into single particle imaging inside cells. So we performed subtomogram averaging on these 194 NPCs, and this is the resulting structure from those subtomogram averaging. It's about 35 angstrom resolution, so nothing to write home about, but good enough to dock in 16 copies of the Y in each of the cytoplasmic and nuclear rings, as well as 8 copies of that NUP93 complex into the inner ring assembly to make up the whole nuclear pore here. So in comparing this structure with other structures that had been published, we noticed a few differences. In the cytoplasmic ring, we noticed these regions with the blue arrows that we think are the NUP214 complex, which is involved in mRNA export pathways. And we think it's NUP214 because of experiments from xenopus and other organisms, but we can't definitively say at the resolution we have that that's NUP214. And we also noticed there's this unknown density here at the NUP85 arms in the cytoplasmic ring. I think that has now been identified potentially as NUP205 in some other high-resolution structures that have come out on bioRxiv in the last couple months. But for us, it was an unknown blob connecting the Y complexes in the cytoplasmic ring. The inner ring didn't look so different to us from anything that had been shown before, other than the overall dimensions of the inner ring, which I'm going to show you in the next slide in greater detail. And the nuclear ring, we also noticed these Y complex connecting densities labeled in the black arrows, as well as this unknown linker density that Martin Beck's group had also identified in an earlier study. So overall, the components of the nuclear pore that we visualized were the same as others had seen before, with a few minor differences. But we did notice a few things that sort of stuck out from our study. And one of them was that we noticed the nuclear envelope membranes. The nuclear membranes were asymmetric at the nuclear pore complex. So this is a cross-section of the NPC, and hopefully you can see that the the cytoplasmic ring makes a steeper angle with the inner ring compared to the nuclear ring. So we have this asymmetric orientation of the membrane at the nuclear pore complex. And we can even see this in the raw data. So these are aligned and averaged NPCs, but you can hopefully appreciate here at the place where the Y complex is attached in the cytoplasmic ring, you can see this much steeper angle in the cytoplasmic, the nuclear membrane at the cytoplasmic ring compared to the nucleoplasmic ring. And if we move to the point where the nuclear Y is attached, you can see that this changes a little bit. And if we move to where there are no Y complexes attached, the nuclear envelope becomes nearly symmetrical. So this is all to say that these rings, the cytoplasmic ring and the nuclear ring, impose structure on the nuclear membranes directly. And this asymmetric structure was something that hadn't really been discussed before in the context of the human NPC. Furthermore, we noticed that the central channel of the NPC in our structure was roughly 80% larger than what had been previously shown. So here I'm measuring the dimensions of the central channel, 57 nanometers in our structure, and the inner ring dimensions, the diameter is about 105 nanometers. Comparing this with what had been observed previously, this is from Alex Van Appen and Martin Beck's group. These are from isolated nuclear envelopes, also from human cell line. But you can see how drastically different the central channel dimensions are. So comparing 43 nanometers with 53 nanometers, and we also see a change in, if you compare those two dimensions and you think about the central channel as a three-dimensional volume, because that three-dimensional channel is actually filled with proteins, you can see that the volume of the channel is so much larger in the native cells than it is in the isolated cells, which just suggests that in the native cellular environment, the central channel is dilated, and whether that's due to the influence of the nuclear envelope or due to active transport, we're not really sure, but this is still a striking difference that we noticed. And as I mentioned, that central channel is not an empty void. It's filled with these FG-rich proteins, which form a hydrogel-like meshwork to accomplish transport. So when we observed this, this was really striking to us, because this new dimension or this dilated dimension for the inner ring could be really impactful for how we understand transport. So we can make morph between the two states. One is the isolated state that Martin Beck's group found in the purified nuclear envelopes, and the other is the native state that we're observing from Cryo-FIB and Cryo-ET. And you can see how each of the inner ring subunits moves apart from one another in the native state compared to the isolated state. And that's how the larger central channel is made. Zooming in on this a little bit, we can even see in our structure, so here on the left, this is from the Cryo-FIB experiments, we can see these peripheral gaps or channels between the IR subunits, which in the isolated nuclear envelope structure, these gaps are totally filled. And so our thinking of this is that these gaps between IR subunits may be important for how integral membrane proteins get through the nuclear pore complex. This is probably best visualized with my hands, but you can't see my hands. But you have to imagine that these membrane proteins going into the inner membrane of the nucleus have to pass through the nuclear pore and both be membrane anchored, and any soluble domains have to go through the NPC directly. So we believe these gaps might be important for how these transmembrane proteins are able to go through the NPC and accomplish both of those things. So if we come back out and compare these two differences, both from our Cryo-FIB experiments and from previous structures from purified nuclear envelopes, we see the difference between these two things was that the inner ring structure, the inner ring architecture has changed so dramatically. And we have some ideas for this, but nothing totally concrete here. We know that in the native cellular context, these NPCs are undergoing active transport. They're connected to the cytoplasm, the cytoplasmic and nucleoskeletal networks, the filamentous networks that run through both of those parts of the cell, versus when you purify nuclear envelopes, there's not necessarily active transport, especially if the nucleus is digested and broken open. And when you do that, you also inherently disconnect it from these filamentous networks. So, our ideas are maybe, our favorite idea is that there's some sort of cellular tension applied to the nuclear envelope through these cytoskeletal and nucleoskeletal networks that mediate this difference in nuclear pore architecture. Now, in addition to looking at the wild type nuclear pores in cells, we wanted to take this a step further to understand a bit more about nuclear pore connectivity and how the three rings of the NPC talk to each other. And so we were involved in a collaboration with Mary Dassa's group at the NIH and with her postdoc Saroj Regmi. So Mary's group was using an Auxin-inducible degron system and tagging various nucleoporin proteins with this AID tag to understand the impact of various nucleoporins on NPC localization, transport properties, and NPC stability. And just to briefly overview how this works, this AID is a proteinaceous tag that, in the presence of auxin, a plant hormone, recruits ubiquitin ligase machinery and selectively degrades your protein of interest. So in this case, Mary's group, the protein of interest here were various nucleoporin proteins, and by tagging them and adding the plant hormone auxin, they could specifically deplete various nucleoporins from NPCs inside living cells. So what Mary's group did was they focused their attention on, at least at first, on the Y complex, which I briefly mentioned before, but this is a protein assembly that has 16 copies in both the cytoplasmic ring and the nuclear ring, and it's a primary scaffold for the NPC structure. Without the Y complex, your NPCs aren't there. So what they did was they tagged various components of the Y complex, including NUP96, which is right at the center of the entire Y complex. And they performed a bunch of experiments to understand pore localization, transport, and stability in these cells. So what I'm showing you is here is a bit of data from their paper, which you can read on bioRxiv. In the control sense, in the control state, they're imaging both the neon green, which is attached to NUP96, and MAB414, which is a readout of the FG proteins inside the central channel at the NPC. So this is another NPC marker. And RANBP2 is part of the cytoplasmic ring of the NPC. So these are control cells. You can see they're getting nice nuclear rim staining for these various factors. And then when they add the plant hormone auxin to deplete NUP96, they see the neon signal is gone, which is what you would expect because NUP96 is tagged with the neon moiety. They see that RANBP2 is all of a sudden gone, which means that the cytoplasmic ring is destroyed on the nuclear pore complex. But they still see 414 staining, which indicates that at least the inner ring and central channel of the NPC is still intact. And they also performed some transport assays showing that plus and minus NUP96 depletion, these NPCs can still transport materials into the nucleus. So from their work, they came up with this model for the sort of interconnectedness of the NPC, and in the case of NUP96 depletion, this is combining work from various different types of experiments, cell-based assays, mass spec, a bunch of things. They came up with this model where if you deplete NUP96, this central component of the Y complex, you lose the cytoplasmic ring integrity, and you also lose the nucleoplasmic ring integrity. And so I thought, this is an interesting tool to understand how the rings are connected to each other. So we should definitely take this strain of cells and see what it looks like. Make cryo-FIB lamella and do cryo-electron tomography. So as in the first part of the talk, I created lamella from these cells that had been NUP96 depleted, and we looked at their nuclear pore architecture. So comparing wild type cells on the left, this is what I showed previously, with the structures of nuclear pores from the NUP96 depleted cells, we found that there were 3 different classes of NPCs in those cells. The first class had all 3 rings, more or less as the wild type structure does. It looks pretty much the same. It measures nearly the same. There's nothing different about it. We found a class of NPCs that did not contain the cytoplasmic ring, which was anticipated based on some of Mary's lab's data, as you just saw with the RAN-BP2 staining going away. And then we also saw a third set of structures which only contained the inner ring. And this suggested to us that the cytoplasmic ring is more easily removed from the NPC than the nuclear ring, because we were not able to see a class of structures that contained the cytoplasmic ring, but not the nuclear ring. And the reasons for that, I don't necessarily have a great answer. It's possible that that ring is more easily degraded because there's more protein degradation machinery nearby, or maybe the spot where the tag was more easily accessible. But regardless, we were able to see this ensemble of structures from a single sample in cryo-FIB and using electron tomography. If we measure these NPCs, we also see something very interesting for understanding NPC biology. So in the wild type sense, you can see each of these dots corresponds to a single NPC that has been measured for its diameter. So each of these black dots here is the diameter of a wild type NPC from the first data set that I spoke about. You can see that the median diameter is about 105 nanometers, but there's some spread to the data. If we look at our individual classes, you can see that the first class, which contains all 3 rings, really has no difference from the wild type data. The second class, which is lacking the cytoplasmic ring, you start to see a bit of an increase in the inner ring dimensions, and we see a little bit more spread. But by the time you get to this third class, which is inner ring only, you can see that the median diameter has increased significantly, and we also have this very large distribution of different size NPCs, some of which are almost 1.5 the diameter of a standard NPC. So they've gotten really, really large. And what this suggests to us is that these outer rings, the cytoplasmic ring and the nuclear ring, act as molecular rulers that define the dimensions of the inner ring architecture. We also noticed that the nuclear envelope asymmetry changed when we removed these rings. So if we're taking, these are cross sections of the wild type NPC, and I described to you how the cytoplasmic side is steeper compared to the nucleoplasmic side. In class 1, again, has all 3 rings. We see that steeper and the CR compared to the NR. But then when we lose the CR, you can see that the membrane collapses. When we lose both rings, the membrane is pretty much symmetrical. And this confirms for us that those peripheral rings, the CR and the NR, do directly shape nuclear envelope curvature. And you can also see in this figure how the inner ring subunits move apart compared to one another in these various classes. So in class 3, that has just the inner ring and it has the largest set of NPC diameters, you can really see that these gaps between subunits are very substantial at this point. And this is how the morphology of the inner ring is able to, or how the inner ring is able to adopt different morphologies. These subunits separate from one another and allow the entire ring to expand in diameter. So our model for this is that a wild type NPC with a 3-ring assembly has a native, what we're calling a native inner ring structure, and it has this asymmetric nuclear envelope. But when we deplete NUP96, we lose those peripheral rings, the cytoplasmic ring and the nuclear ring. And what that does is it allows the inner ring to dilate because there's no longer a limit posed based on those rings. There's no longer a limit posed other than when the NPC would fall apart, more or less. The inner ring can dilate as large as it wants until it can no longer make the necessary interactions to hold itself together. So the CR and the NR are acting as the rulers that define the central channel shape. So the fall of last year was a really exciting time for the NPC. I feel like I have to show this slide because our paper came out. Sorry, I realize I have the dates messed up here. It's not 2020, it's 2021. But our paper came out from human cells, and then Christian Zimmerli and Martin Beck published a paper where they also noticed these IR dimension fluctuations between various states of S. Pombi. Again, I apologize, I have the date wrong. And then Chris Akey and working with Mike Rout and Liz Villa did the same thing, comparing isolated structures from Cerevisiae, and they saw that the isolated NPC had a smaller inner ring compared to the NPC studied directly in situ. They also see the same gaps that we saw. So this was all really awesome to be able to see this across various cell types. And then even still, Shamal Mosalganti from the Beck group, he now has his own group, Michigan, published a similar thing from more human cell types and was able to use AlphaFold to create a more complete model for the NPC in its entirety. But it was a really exciting time to be able to see this phenotype in understanding how the inner ring of the NPC can change its architecture across these various eukaryotic cells, all around the same time. So I just want to wrap up with a slide here to say that I think the future of structural biology is here, and I'm really excited to be a part of this. So for me, I want to understand the precise mechanisms of how the inner ring is rearranging to allow for this central channel change, and what the inner ring dilation really means for things like cargo transport, both in terms of rates and selection. I didn't mention, but I will verbally, that Martin Beck's group saw that whole HIV-1 capsids can be transported through these NPCs with a dilated inner ring configuration. So there's definitely going to be a lot of interesting biology to follow up on here to understand, what exactly the NPC is doing at various states, and whether or not various states are NPCs in a single cell can adopt these various states all at the same time, or if it's sort of a continuum. rather than like an open and closed. I think there's a lot of interesting biology here to follow up on. The other thing that is exciting me about electron tomography is doing what people are calling visual proteomics. And that's primarily because in these tomograms, there's so much to look at. You know, you can stare at these things for days and identify new particles. I'm sure anybody watching can probably pick out a few things that I haven't even noticed in this tomogram as it's flying by. I think we're at a point where we're going to be able to take this data and try and figure out what we do know, solve structures of complexes we do know from this native cell data, and also hopefully try to find what we don't know in this data by computationally searching for molecules throughout the tomogram space. And the thing that maybe excites me the most about Cryo-ET is incorporating cellular context with high resolution single particle structures. And that's just because, you know, the cell is this crazy, messy place. Well, I took this image from Digizyme, and the cell is this crazy, messy place. It's not the ideal cryo-EM grid with just your protein of interest on it. So I think understanding how various proteins talk to one another or exist in the cell in their native environment is the most exciting, to me, the most exciting avenue for Cryo-ET in the future here. So with that, I have lots of people to thank. I know we're getting close to the time, so I'm going to fly through this. Obviously, I have to thank Tom Schwartz, my mentor back at MIT, here for allowing me to take on this crazy project when you know, our microscopes were just being built at MIT. When I started my postdoc, we didn't have any electron microscopes. And when I ended, we had 2 electron microscopes and 2 cryo-FIBs and a bunch of more toys on their way. So it was a really fun time to be there and sort of be at the cutting edge of all of these techniques. I have to thank, obviously, Ohad Medalia's lab. They were our early collaborators in this NPC project, specifically his student Matthias, who helped with all of the subtomogram averaging, as well as Matthias probably should be credited with teaching me how to do electron tomography in the first place. It was my first tomogram that I collected next to Matthias at Ohad's Polara in Zurich. Thanking Karsten, who was really helpful in understanding all of the NPC biology with this story, and he also co-mentors Matthias. Mary Dassa's lab at the NIH, specifically Saroj, who worked on the Auxin-inducible degron cell lines, and that unlocked our understanding of how the NPC rings are interconnected. We had 2 great facilities at MIT. The MIT.nano cryo-EM facility, where we did our TEM and our cryo-FIB imaging, which is led by Ed Brignall. And then the Koch Institute Nanotechnology Core housed the Zeiss cryo-FIB instrumentation that's led by Abigail. So I was a very popular user in both of those facilities. And then finally, the Nogales lab, I moved to Eva's lab in December of 2020, mid all the pandemic mess, and she's taken me in. I've started some new projects in the Nogales lab, none of which I have talked about today, but maybe I can talk about at some point. And obviously I have to thank the funding from my foundation and NIH. And there are some Twitter handles if anyone wants to follow us on Twitter. Otherwise, thank you for your attention, and I think the hill is going to break back in here.
Thank you very much, Anthony. It was a beautiful talk. Just as a humble reminder to the audience to please submit your questions by typing in. It is a question panel that you see on the right-hand side. Just a heads up, due to limited time, several questions will be taken offline. So the answers to your questions will be sent by emails. So Anthony will be taking just maybe a few questions since we are limited on time. So the first question is that there are several questions about water faceplate. Have you tried using faceplate and tried in-focus imaging TIL series? If not, what is the defocus that you use for your TIL series acquisition?
Yeah, that's a wonderful question. So I avoided the Volta faceplate, mostly because I had heard from colleagues that it was kind of challenging to use. So all of this data was collected without a faceplate. The defocus varied between -2 and -5. But I am very interested in how faceplate technology could help electron tomography. I know there are some groups working here at Berkeley, and I think some others across the world working on laser faceplate technology, and I'm really, really curious to see how that develops for improving tomography data collection. But I did not use a faceplate.
Thank you, Anthony. Another question, have you tried varying the slit width of your bioquantum system? If yes, what slit widths did you try?
I'm sorry, I missed the beginning of that. Have I tried varying the what?
The slit width, because you use 2080 slit width for the energy filter. And if you tried using or varying the slit width, was it any difference in achievable resolution of the TIL series?
Yeah, I very early on stuck with 20. I tried 10, inspired by someone else's paper, but what I found was I just wasn't able to collect as many tomograms without needing to recenter frequently. So I just very early said I'm going to go with 20 because I can collect 8 or 9 hours of tomograms without needing to recenter this. So it was purely dare I say, a laziness thing. But I guess I don't have the perfect answer for this question because I've stuck with 20 for 99% of my collection time.
Okay, thanks, Anthony. Another question, and that will be the last question. The central channel dimensions could be different due to cell stress. Is it possible or difference between the wild type cells versus the HeLa cells? Also, wild type cells are more susceptible to changes in the buffer condition or the growth media condition. Is that possible?
Yeah, so I'll address that in two parts. So we in our paper we looked at a couple cell types, and I think we reported two of them. The central channel dimensions are pretty similar between the varying cell types that we looked at least. We more or less thought them to be the same. So anything that was done anything that was looked at via Cryo-FIB was the same. Now the question about cellular stress, I think, is a really great question. So Christian Zimmerle's paper in Pombe cells is all about energy depletion and membrane tension, and I think that there's some potential, as this question has provided, that cellular stress would impact the membranes in the cell, or impact the cytoskeletal networks, or impact any of these molecules that touch the nuclear envelope. So I think that it's very likely that cellular stress can impact the in-ring, the channel dimensions, and we're just going to need to more precisely figure that out. But I would suggest that this person look at the Zimmerle et al. Paper from Pombe, because he spent a lot of time looking at how energy depletion via nutrient changes in media for Pombe, at least, affected nuclear pore morphology. I'd love someone to do the same things in human cells.
Thank you, Anthony. Rest of the questions will be taken offline. Again, thanks, Anthony, very much. Thank you, everyone, for attending the talk. Have a great day, Anthony. The final note to you.
Thank you.